Vector, epidemiology. In epidemiology, a disease vector is any agent which carries and transmits an infectious pathogen into another living organism. Most agents regarded as vectors are organisms, such as intermediate parasites or microbes, but it could be an inanimate medium of infection such as dust particles. Arthropods. Arthropods form a major group of pathogen vectors with mosquitoes, flies, sandflies, lice, fleas, ticks, and mites transmitting a huge number of pathogens. Many such vectors are hematophagous, which feed on blood at some or all stages of their lives. When the insect's blood feed, the pathogen enters the bloodstream of the host. This can happen in different ways. The Anopheles mosquito, a vector for malaria, filariasis, and various arthropod-borne viruses, arboviruses, inserts its delicate mouthpart under the skin and feeds on its host's blood. The parasites the mosquito carries are usually located in its salivary glands, used by mosquitoes to anesthetize the host. Therefore, the parasites are transmitted directly into the host's bloodstream. Pool feeders such as the sandfly and black fly, vectors for pathogens causing leishmaniasis and onchocerciasis respectively, will chew a well in the host's skin, forming a small pool of blood from which they feed. Leishmania parasites then infect the host through the saliva of the sandfly. Onchocerca force their own way out of the insect's head into the pool of blood. Triatomine bugs are responsible for the transmission of a trypanosome, Trypanosoma cruzi, which causes Chagas disease. The triatomine bugs defecate during feeding and the excrement contains the parasites which are accidentally smeared into the open wound by the host responding to pain and irritation from the bite. Plants and fungi. Some plants and fungi act as vectors for various pathogens. For example, the big vein disease of lettuce was long thought to be caused by a member of the fungal division Chytridiomycota, namely Elpidium brassicae. Eventually, however, the disease was shown to be viral. Later it transpired that the virus was transmitted by the zoospores of the fungus and also survived in the resting spores. Since then, many other fungi in the Chytridiomycota have been shown to vector plant viruses. Many plant pests that seriously damage important crops depend on other plants, often weeds, to harbor or vector them. The distinction is not always clear. In the case of Bixenia graminis, for example, Berberis and related genera act as alternate hosts in a cycle of infection of grain. More directly, when they twine from one plant to another, parasitic plants such as Cuscuta and Cassitha have been shown to convey phytoplasmal and viral diseases between plants. World Health Organization and vector-borne disease. The World Health Organization, WHO, states that control and prevention of vector-borne diseases are emphasizing integrated vector management, IVM, which is an approach that looks at the links between health and environment, optimizing benefits to both. In April 2014, WHO launched a campaign called Small Bite, Big Threat to educate people about vector-borne illnesses. WHO issued reports indicating that vector-borne illnesses affect poor people, especially people living in areas that do not have adequate levels of sanitation, drinking water and housing. Vector-borne zoonotic disease and human activity. Several articles, recent to early 2014, warn that human activities are spreading vector-borne zoonotic diseases. Several articles were published in the medical journal The Lancet, and discuss how rapid changes in land use, trade globalization, and social upheaval are causing a resurgence in zoonotic disease across the world. Examples of vector-borne zoonotic diseases include: many factors affect the incidence of vector-borne diseases. These factors include animals hosting the disease, vectors, and people.